Students, today we will study virus. Virus in Ivanescu in 1892 found that an infected extract from the tobacco plant with a mosaic disease was still infectious through filter that retain almost all known bacteria. He assumed that the infectious agent was a small microorganism. In 1898, Benzernik obtained a similar result and called the fluid as a contagion virus fluidium or contagious living fluid. Later, he observed that the infectious agent was too small to be visible even under microscope and unlike bacteria could not be grown in a cell-free medium. They came to be known as a filtrable viruses. In the early 1900s, a distinction was finally made between bacteria and virus. About 2 to 3 decades, other important diseases of both plant and animal were shown to be caused by a similar infectious agent. With the passage of time, these observable filterable was gradually dropped, and the word virus, derived from the meaning poison, became a specific group to designate all such ultramicroscopic infectious agent that cannot be grown on a non-living medium, pass through bacteria retaining filter, and responsible for many infectious diseases of man, animals, and plants. In 1950, F.W. Todd and F.D. Harrell independently noticed that the bacteria were also infected and destroyed by similar agent. Such viruses that infect only bacteria were named bacteriophages by F.D. Harrell. Perhaps the most important combination in the contribution in the field of characteristics of these agents was discovered by Wendell Stanley in 1935, who showed that the tobacco mosaic virus can be crystallized, making it possible for the first time to carry out chemical and structural studies on a purified virus. It was also found that the non living crystalline material could infect the healthy plant and replace itself. At first, the investigation showed that virus consists of protein. In 1936, Badgen and Perry demonstrated that the tobacco mosaic virus consists of 95% protein, 5% nucleic acid. They established that all the viruses were principally composed of a nucleoprotein. With the advent of the electron microscope in 1940, it became possible to study the morphology of virus. We now know that the viruses are found as an obligate intracellular parasite in all organisms. Viruses infect the fungi, bacteria, and protista as well. They can reproduce only within the cell. They are the major causes of disease in human. Some diseases of agriculture importance, animals and plants are also known to be caused by viruses. A further advance in the virology was made in 1959 when a virus infection was found to result in the production of a group of protein known as interferons. One of the interesting property of the interferons is that it is not a virus specific, rather it is a cell specific. It is secreted in the exceedingly small amount by the virus infected cell. It is by itself not active against viruses, and after leaving the cell in which it was produced, its protein neighboring uninfected from the viral infection. The term interferons has been given to this phenomena. Since then, the possible therapeutic value of the interferon is being investigated to produce the immuning vaccine. They are widely used as a biochemical tools by the molecular biologists. Recently, beneficial effects of virus have started to be explored. It has been indicated that the virus may have an exciting and important role in gene therapy, inserting missing gene or replacing a defective one in the human cell. This technique uses a harmless virus to carry the missing or the new gene into the certain host cell where the gene is picked up and inserted into the appropriate chromosome. General character of virus says, although this group is a very extremely heterogeneous group, and the viruses have shared certain properties. They are the smallest simple of all known organisms. They can also only be seen with an electron microscope. They are able to pass through filter that prevent the passage of most of the bacteria. Viruses cannot be cultured on any synthetic media. That is, they are acellular or non-cellular particles that completely lack structure characteristics of cell. A striking feature of the many viruses is that they can be crystallized. Virus crystals dissolved in water can initiate an infection cycle. They lack the cellular machinery necessary for acquiring nutrient, producing energy, and synthesizing protein. Viruses are absolutely required by the living host cell to perform nearly every biological function necessary for the survival, and thus they are described as an obligate intracellular parasite. They are incapable of an independent metabolism, a property that distinguishes them from certain bacteria such as rickettsias and chlamydias, which are also very small, filterable, and obligate intercellular parasite. Virus infect every kind of organism on the earth, that is plant, fungi, protozoa, insect, birds, mammal, and human beings. Even bacteria are attracted and killed by viruses. These bacterial viruses are called bacteriophages, or simply we call it as phage. Viruses which attack fungi are called mycophages. Each virus particle consists of a core of genetic material, that is the nucleic acid, that is covered by a protein coat called as a capsid. They consist of nothing more than nucleic acid and protein. A very characteristic feature that is peculiar to only virus is that their genetic material consists either DNA or RNA. However, never both. The genetic material of all other form of life contain both DNA and RNA. The complete mature virus particle is referred to as viron. Most viruses are highly specific for particular organism. They can ordinarily invade cell of one type, example plant, animal, and bacteria. Often a single host species or even strain within a species, and only one tissue or organ in that species. Like other organisms, viruses are capable of self reproduction. But the process of virus reproduction is different from that of other organisms in the sense that viruses do not divide to form a new individual. Without exception, viruses only multiply inside the living host cell by using the synthetic machinery of the cell. The method of virus multiplication is called replication. Viruses have an ellipse phase in the life cycle. While producing in a host cell, the virus particle disintegrate into its molecular constituent. The eclipse phase ends with the reappearance of progeny virus. Viruses are easily transmitted from one organism to another by mechanical means or by biological vector. Viruses are the causal agent of highly infectious disease in a wide variety of organisms. They causes severe economic damage to several crops. Viruses are also severe human disease such as the infectious hepatitis, poliomyelitis, herpes, and AIDS. All the viruses are generally insensitive to the broad range of available antibiotics such as penicillin, streptomycin, and other. Thank you.